Welcome to this video on Translational Geroscience. We will focus on cutting through the noise of the hallmarks of aging to identify empirically validated clinical targets, creating an evidence-based prioritization roadmap for biomarkers and therapeutics. Translational Geroscience is accelerating from a theoretical framework to clinical validation. This timeline shows the evolution from the original 9 hallmarks of aging in 2013 to the emergence of age signatures and the expanded 12 hallmark framework by 2023. It is important to distinguish between lifespan and health span endpoints. Lifespan focuses on time to death, while health span measures time lived free of major disease and disability. The core metrics and limitations differ significantly between the two. The 2023 expanded framework now encompasses 12 interconnected processes. Disabled macroautophagy, chronic inflammation, and dysbiosis are 3 new hallmarks, reflecting current research findings and the need for standardized human assays. This flowchart illustrates the mechanistic bridge from biological processes to measurable clinical endpoints. It moves from the 12 hallmark processes to human biomarkers, risk stratification models, and finally to the terminal endpoints of longevity and health span. Integrative biological age readouts consistently outperform single hallmark markers. The strongest correlations with health span come from measuring downstream integrators, such as composite clocks and systemic inflammatory signaling. The mortality risk spectrum highlights inflammation and mitochondrial dysfunction. Low mitochondrial DNA copy number and high IL-6 levels carry massive independent mortality risks compared to standard proteomic acceleration measures. This human evidence matrix highlights the readiness of systemic and epigenetic targets. Hallmarks like epigenetic alterations and chronic inflammation show strong correlations with both longevity and health span, and have high assay feasibility. Translating cellular biology to human populations remains hindered by assay limitations for certain hallmarks. For example, disabled macroautophagy and stem cell exhaustion currently lack broadly standardized human assays. The intervention reality check shows that target engagement often outpaces functional validation. While interventions like senolytics and caloric restriction show proven target engagement, functional outcomes often remain unspecified or complex to interpret. The category overlap problem shows why correlations diverge across hallmarks. Clinical cohorts often measure composite biological signals, blending elements like senescence signals and ECM remodeling, leading to high predictive power but reduced mechanistic specificity. The clinical prioritization roadmap for near-term geroscience points to chronic inflammation and epigenetic alterations as having the highest translational potential, based on their strong mortality effect sizes and validated risk stratification capabilities.